My name is Titus-McQuillan from the University of Texas at Arlington. And today I'm going to do a talk focusing on some of the gene flow that we're seeing in the Australian gecko, Heteronotia binoei. This is a primer to some of the work that I've done in my dissertation, looking at gene flows, a primer to understand diversification, or at least the mechanisms and processes that will promote diversification or limit diversification and could very well lead to speciation. So we're focusing here in Australia. So as a primer, Australia is a great place to look at gene flow and understand the dynamics of diversification. Broadly speaking, if you take a look at Australia, it's broken down into four major biomes. You have a monsoonal tropic biome in the north, you have an arid biome in the middle, and then you have these amazing biomes, a southern one to the southwest, and an eastern one to the east. Now these biomes aren't homogenized, they're not only in four environment types. If you take a look at this map here, this is foliage variation cover over the whole continent of Australia. You'll see that it actually is quite varied. So you end up having quite a bit of environmental types and habitat types that allow for a lot of local adaptation or just a bunch of diversity to occur between all the organisms within Australia. And to kind of fortify that a bit further, what we see here is the climate and the precipitation of Australia. So as you move across the continent, you'll see that there is variation influencing these different habitat and environment types. And another very important dynamic of Australia diversification is within this last 100,000 years. What you'll find is that Australia wasn't always this dry place. Australia is the second driest continent on Earth, but it had times of being wetter in the past. So you have these fluctuations between wet and dry periods due to glaciation. Now, Australia didn't have like a whole ice sheet over it, like you see in maybe Europe or North America during the last glacial maxima. What instead happened is that water was contracted into ice, and in Australia, you ended up getting this aridification event. And so you have this ebb and flow of wet and dry periods where refugia of wet environments ends up contracting, and you have this new arid expanse of new niche that many of the organisms can then flow into and diversify, and you have this expansion and contraction within these refugia, and so that's how you get the dynamics at play. And you find that Australia is very diverse within a number of animal groups. You'll see it in marsupials, you'll see it in reptiles and amphibians, insects, and even many plants like Australian acacia and eucalyptus. Today we're going to focus on these cute little geckos here, Binoei's gecko, Heteronotia binoei. This is a great system to study in Australia to understand the dynamics of diversification. If you take a look here, this is a qualitative map showing the distribution of Heteronotia colored by all the different lineages. And what you see, there's a lot. They cover nearly all environment types throughout all of Australia, and they're very, very diverse. So Heteronotia is a known species complex with many more than probably one species, but if you look at how complicated this is, it's very hard to tease out what should be considered a species and what needs to be split. So to narrow it down, we'll focus up here. This is the Australian monsoonal tropics. These are all data points that have genetic data to them, and you see it is very, very diverse. It's a complicated system to look at because there are so many different lineages and variation and dynamics going on across environment and across the genetic spectrum. But that also makes it a really good system to study, simply because you can look at many different lineages and understand the dynamics of diversification and how and their interplay between them. We're going to focus in Queensland here. This is where my samples were, so you can take a look here. Now, Queensland, this area of Queensland is a really good spot to study dynamics of gene flow because this area here has an amount of lineages that have some deep splits in the phylogeny. So we're going to look at some putative hybrid zones. These are transects of continued distributions where you have different lineages overlapping with one another, and each of these boxes that you see in putative hybrid zones are at different varying degrees of divergence. And so we also have replicates throughout, so you can tease out the dynamics of gene flow between shallow divergences and deeper divergences, and see if you have promoted gene flow or if you have limited gene flow, which may very well lead to what would be a speciation event. And the colors come from the phylogeny that you see here. So my simple question for this talk, just to kind of look, is there gene flow between these lineages that we see in Queensland? And so my approach to doing this was getting genomic data using target capture of the exons. We're looking at around 3,000 exons throughout this time. Now, if you take a look at this density here, you can see this deeper divergence between the lineages. So you have this western group, and you have this eastern group, and you have a deep divergence between them. While in between these groups, you end up finding that you have a much shallower divergence. So you can play with the dynamics because if you look along, say, this Gulf lineage here, you have replicates ranging across the zones, and so you can test to see if deeper divergence leads to more, or at least less gene flow, and the opposite, if you have shallower divergences, you end up having more gene flow. And so to start out with, we have a DAPC plot right here. And what you can see is what we'd expect. We have clusters of lineages that are more closely related, being closer to one another. One thing that is something to look at is we have a cluster here of this Gulf lineage. This Gulf lineage is pretty well distributed across the land. So if we take a finer scale look and look at these transects, what we'll find is that structure is giving us what's giving us structure is looking at there could be gene flow between this group of CY6 broken down into its smaller sublineages here. So in order to test that, I did an AMP-VAVA test using EVO by R by Heath Blackmon. And what we see is we get a positive D statistic, which leads us to estimate that there is actually gene flow in between these taxa here, this EA6 and CY6 south group. So we do see gene flow within these shallower divergences. Now, if you look at if you look at hybrid zone 2, you'll find pretty significantly there isn't any gene flow in the structure plot. It's kind of boring. You just get a green cluster, a green block, and a red block between EIU and this Gulf lineage. However, I think this is a pretty significant result. If you know that there's no gene flow going on, this could very well be a speciation event. And then finally, if you take a look at hybrid zone 3, what we'll find is we have what looks to be structure and gene flow in between. And so we test that again, an AMP-VAVA test. This time, we're getting a bad result, this significant negative D statistic. And what we find is that this integration, we can statistically say is significant if there is some sort of gene flow going on between these groups. And so here again, you have some moderately shallow divergence in that's leading to gene flow between these different lineages. So to conclude, what we're seeing here is that some of the more shallow divergences between your CA6 groups and this NWQ and Gulf group down here, you're finding that there is gene flow happening. But as you get into overlap between deeper diverged lineages, you'll find that there isn't any gene flow. And I think this is one of the cool results that we'll find here and we should look more into, being that something's at play here where you have these lineages no longer having gene flow that are in contact with each other. So I would like to further investigate if it's some sort of environmental process, if it's physiological, some sort of genetic process, who knows? Do all the analyses. So with that, I would like to thank the Fujita and Moritz labs, all my UTA graduate students who helped me and support me through this, my funding sources, ANU and UTA for having me, and a special thank you to Matt Fujita and Craig Moritz for giving me insight into this very interesting and complex group that we're working on. And thank you.